said yesterday, we are looking at functional genomics, computer analysis. Functional genomics, computer analysis. What this simply means is that, what is in simple terms, this means that we are going to be looking at molecular biology using computer. So we are going to be using computer instead of just staying in the lab and doing some work using PCR apparatus. So in this aspect, it's computer that we use, not that we just go to the lab, add this to this, or any other using the machine. The only machine we use is computer. What this means is that we are going to be using databases already, what have been produced, sequences, protein, everything that is on the net. That's what we're going to use and compare. And this is an area that many people in advanced countries are going into now. When I was in the US, about 2015-2016, during my Fulbright fellowship, there was a particular lab. They are Egyptians. The husband is an Egyptian, the wife is an Egyptian. So the two of them are environmental and biologists. They plan for Bioinformatics, which is also part of functional genomics, computer analysis. So they are focusing on bioinformatics. They left the wet lab. They go to wet lab. In wet lab, you know wet. When they say something is wet, dry lab, this is dry lab. Don't go to the lab, we are doing lab work. So which one is easier? Is it to go to the lab looking for all this one? I was in India 2 years ago, I came back December last year. You know, I left December 2 years ago, I came back August last year. And I went to a conference at one of the state universities. And then different posters. So as foreigners, they gave us privilege to speak at the conference as speakers. At that conference, I spoke to them about the background. And another conference, I spoke to them about that. So, you know, they want to have foreigners be part of the conference, not just Indians. When they see us, they want to maximize the opportunity. So we are invited. So I was to score the poster at the conference, part of the recognition. So I was going around. The lady, I saw everything she did. I said, please, did you go to the lab? She said, no. I now said to her, I said, and you have the results. So everything was on the computer. Everything was on the computer. And that is the future of biology. It's what this does is that it shortens. You don't need to waste your time in the lab doing trial and error, optimizing. You will have optimized on the computer. The computer will have doing the test. You just go and use the test in the lab and produce. This was what was used during COVID. During COVID, people were down. There was no time to be testing and testing. Normally, when you want to develop a drug, it takes years, or vaccine, it takes many years, sometimes 10 years to be tested. You have to do this, check that, check it with animals, check it with humans. But in this case, there was no time. People were dying every day. So they used this to produce one of the vaccines. I think Pascal, very cynical, or something like that. This was what they used. So just go molecular docking. You do the docking, get the results, and then you flash it out, and then you produce the vaccine. Though definitely to have its own side effects, but at least it's able to save people to some extent. Let's write it down. Functional genomics is a branch of genomics. Functional genomics is a branch of genomics that focuses on understanding the functions and interactions of genes and proteins that focus on understanding the functions and interactions of genes and proteins. Functions, interactions, genes, proteins, of genes and proteins. Other than just their sequences. Other than just their sequences. So this is not just about sequencing. It goes beyond sequencing. You just sequence the blood, this is the name of the sequence. Like I tell my students, some time ago we used to do the research of who is there. That's how the research we do those days. Who is there? Who is there? We want to check the microorganism in the soil of that place. What the microorganism? We want to check the microorganisms. We want to check the microorganism in the water at that place. We want to check the microorganism in the air of that place. Research has moved now. The microorganism in that air, what is it doing in the air? Of what use is it? What can we use it for? That's where we are now. The microorganism in the soil. Don't just tell us that it's just microorganisms in the soil. What can we use them for? Beneficial. That's the application. So, at another point, you now say, okay, we are going to know PCR. Anyone that knows PCR, he has done good research. Those days, when they had banned, the PCR banned, people just go and get PCR banned, and they say they have done PCR. Whereas that's not the PCR. When you show the PCR banned, you didn't show the sequence of what uses it. A particular student many years ago, she did a particular project. She was to work on E. coli from the start to finish. The topic was E. coli. And then she said she sequenced. She said she was doing E. coli. Then I wanted to write the paper. And I took it. I was in the US then. I just took it. And the other time, I didn't know how to blast. While I was there, I didn't know how to blast. I didn't know how to blast. She came, she went to do the research, she said, it's E. coli. And then we said, it's E. coli, it's E. coli. Me, I don't know how to blast. That there's something called blast. After you have sequenced, you blast. Basic local alignment /2. By the way, this question was asked me, like, what is the full meaning of blast? What is the PhD defense? They know. They are actually blasted it, so you blast it. When you have the sequence can be ATC, CT, A, something like that, continuation like that. That sequence, very long. Yes. The next thing, that's just a sequence. After you do PCR, then you go and sequence. After you sequence, this sequence you have, then you use the basic local alignment /2 to compare it with what is already existing. That's just the game, that's what you are doing here. So I now blasted that the sequence she said she got for E. coli. It was proteus. It was proteus. So I was in the US, I already know how to blast. It was proteus. You're laughing. The topic was E. coli. So all the tests, so I look at, okay, let me see if I can use proteus to write the paper. And I went back, everything from introduction, my total method was E. coli. So I said that just their sequences. Full stop. With the explosion of high-throughput technologies, with the explosion of high-throughput technologies, such as next-generation sequencing, NGS, high-throughput technologies such as next-generation sequencing, NGS, microarrays, and proteomics. Microarrays and proteomics. Platforms. With the explosion of high-throughput technologies such as next-generation sequencing, NGS, microarrays, and proteomics platforms, computer-based Continuation. Computer-based, in bracket, bioinformatics. Computer-based, bioinformatics. Bioinformatics. Computer-based, in bracket, bioinformatics. Analysis has become essential for interpreting large-scale genomic data. Now, the topic we're looking at is functional genomics, computer analysis. Now, this is we'll just define functional genomics. In the process, I spoke about interaction of genes and proteins. So, what's the difference between gene and protein? A gene is a DNA segment providing instructions. A gene is a DNA segment providing instructions. Protein is a functional molecule built from those instructions. Protein is a functional molecule built from those instructions. So the gene provides the instruction. The protein is what is built from those instructions provided by the gene. So the gene is what is providing the instruction, while the DNA is what is the protein. The protein is a functional molecule built from those instructions. So when you have a gene that codes for this, you will not have a protein. So that's the protein. So it's the gene that points direction of what the protein. Functional genomics computer analysis involves functional genomics computer analysis involves the use of computational tools. Involves the use of computational tools, databases, and statistical methods. Databases and statistical methods to analyze gene expression. Gene regulation, protein function, and biological pathways. So that's functional genomics, computer analysis. We use computational tools, databases, statistical methods. You can see this one does not involve you going to the lab, but you must have knowledge of the laboratory. So you can't just wake up and just say, I'm not going to do anything, I just want to go and do computer genomics. You don't know anything about biology, about biochemistry. Data types and functional genomics.
Before the last one, protein function and
biological pathways. Protein functions and biological pathways. Data types and functional genomics. That's types of data. Number one, gene expression data. Number one is gene expression data. This is generated using microarrays or RNA sequencing. This is generated using microarrays or RNA sequencing. It measures the level of gene transcription under different conditions. It measures the level of gene transcription under different conditions. It is used to identify unregulated. It is used to identify unregulated and downregulated genes. It is used to identify unregulated and downregulated genes. Number 2 is epigenomic data. Epigenomic data. Epigenomic data. This includes DNA methylation. This includes DNA methylation, histone modifications, and chromatin accessibility. This includes DNA methylation, histone modifications, and chromatin accessibility. Epigenomic data is generated using techniques. Epigenomic data is generated using techniques such as chip sequence, CHIP sequence, and also ATAC sequence. It helps in understanding gene regulation. Number 3 is proteomic data. Proteomics data. This studies protein expression. Protein structure and interactions. Studies protein expression, structure, and interactions. It is generated through mass spectrometry. It complements transcriptomic data. Number 4 is functional annotation data. That's number 4. Functional annotation data. This describes gene functions, pathways, and biological roles. As a function annotation type data describe gene functions, pathways, and biological roles. It is obtained from curated biological databases. Biological databases. Now let's look at the key computational tools and software. Number 1, sequence analysis tools. And among the sequence analysis tools is BLAST. The A here is BLAST, Basic Local Alignment Search Tool. Sequence analysis tool. The first one there is BLAST. BLAST identifies sequence similarity. And predicts gene function. The second one, which is B, is cluster omega, in bracket MOSU, or stroke MOSU. Cluster omega, then stroke MOSU. MUSCLE. The MOSU should be for capital letter. Cluster omega. The cluster should be AL, it's not CR. AL. Cluster omega /MOSU. This helps with multiple sequence alignment. This helps with multiple sequence alignment. So we're looking at the key computational tools and software. The first one is what? Sequence analysis tools. The second one is gene expression analysis tools. Gene expression analysis tools. The first one here, which is A, is capital G, capital E, capital O, 2R, GO2R. 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 Online analysis of gene expression datasets. So that's what this GO2R does. Online analysis of gene expression datasets. I will try to ask us to do some things because this is actually a practical. What's really practical? We have a computer. Everybody has a computer, and then we are checking some things, checking these different databases. And you see, like they say, the internet will not forget. So anything you put on the net is going to. I was in India, and then we were talking about some things. The student came to me and I told her, oh, you have to do molecular analysis for your organism. So I was trying to encourage her. When you do molecular analysis, NCBI, all those things, I was explaining to her. And I now said, in fact, you can get new organism that will be on in the gene bank. Yeah, and I said, okay, as I was just explaining and showing her the a blast NCBI meeting. And I said, me, I have some organism now. They deposited the database. As I was explaining, I was online. I just pressed my name inside this thing. It just brought all the organisms that deposited. I said, can you see now? See what I'm saying now? You can see my name here. 4 organisms. I never even knew that I could just check and find it. So 4 organisms. So I just said, so you can see now. These are what I'm talking about. And because of lack of understanding of some of these things, these functional genomics analysis, many opportunities that we have in our research here, we don't have, we just leave it at the crude stage. We leave our research at the crude stage. Crude. Just leave it there. We don't know that after the lab work, the work just started. That you will do something, you will design some things, you will say, Before I went on Fulbright scholarship, I read a lot. I've heard long ago that when you want to do research, you should read about 30, 40 papers in the area where you want to do your research. When your research wants to be on soil rhizosphere, you should read 30 different papers on soil rhizosphere so that you are starting with information, so that you're not starting with nonsense. So when you know those 30 papers, you know what is on the what you got, what is on the what you got. You cannot decide, okay, this is what I want to build on what this guy did. This guy used this guy used this guy used this. You have seen the trend. You will not go another direction. So all the 40 papers are brains. You will not crystallize it and see area that are not focused on. You will not focus on that area. You will not focus on that area. So I used to read, I read. I downloaded. If I check my list, I'll see. So then if I printed, pay for printing, download, back, print it, I have number 1, number 2, number 3, number 4. Write it like that, staple it. Then, based on that, I have series of, there's no thing we want to use and they will abuse it in their work. There's no figure, they will abuse it. So you have this figure, I like this figure. That's the figure you're going to use. Your data is different. So it's not like just do this, just put it in one table like that. Because I've read so many. The one I used on my seminar for papers I wrote before my defense was different from the one I used inside my thesis. The figure. I had a figure like this. figure was like this, and then like this. So there's something on this axis, there's something on this axis, there's something on this axis. So as something was going like this, another one was going like this. Somebody saw it when I was applying for food, like he said, is it here you did this work? The person is a student that I've gone on food that come back. So I was showing him my document, he's helping to look at it. He said, this work, is it here you did it? I said, yes. It's not about you. It's not about, you can work, I don't know how to put it. It's, this is what they will see. They will not see your lab. Nobody will see your lab. Except if you decide to take a picture and put it in the list. This is what they will see. Your figure, your tables, and other things that you show. The interpretation of your data. So I use it. Normally, it's where X axis and where axis and X axis. But which one be this side again? It's because of the material I used. It's because of the protocol I'm using. So that was that. But eventually at the end, I separated them. There were three things. I can't remember the third one now. There were three different things I was measuring. I was measuring the pH. I was looking at the pH. I was looking at the optical density. Then I was looking at the total material count. So the pH, optical density was here. I can't remember which one. We go at the total material count, and then the pH was here. There are three different parameters that was measured. So the third one, because of the scale, will not be here. So it has to be there. This was like what I was using before, all this thing before the defense. When I was writing the thesis, I separated them because I have control, I have this thing. So this one as it is now, this will control, and I have another one to do the test. I separated it. Instead of having 2, I now have 6. So I have one like this, another one like this, another one like this. Take me 1, 2, 3. Then I have another one control 1, 2, 3, then 6. Again 6. But they were in on one page. I put them like that. In one page. I have plenty. If I put it one per page, it will take up the space. So where are we? I said on analysis of gene expression data sets. Then we also have, under that gene expression analysis tool, we have, we have capital D, capital D is sequence 2, D is SEQ2, D is SEQ2 slash HR, H, EDG, capital R. This is used for differential gene expression analysis. Differential gene expression analysis. Then we also have R/bioconductor. 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 These are statistical analysis. These are for statistical analysis and visualization. For statistical analysis and visualization. Now let's look at, we have looked at sequence analysis tools, gene expression analysis tools. Let's look at functional annotation tools. Functional annotation tools. Functional annotation tools. The first one under the functional annotation tools is David. David. That is rated David. Functional annotation and enrichment analysis. The first one is David. Capital D, capital A, capital D, capital I, capital G. Functional annotation and enrichment analysis. The next one is KEG. K-E-double-G. K-E-double-G. What is this one used for? Pathway analysis and metabolic mapping. Pathway analysis and metabolic mapping. Then we have the third one, gene ontology. Gene ontology. Geneontology. That's G-O. Gene ontology, G-O. Classifies genes by biological process. Gene ontology classifies genes by biological process, molecular function, and cellular components. Molecular function and cellular components. So we are still looking at the tools. The next tool is network and pathway analysis tools. Network and pathway analysis tools. So there are network and pathway tools. The first one here is string. S-T-R-I-N-G. Capital. S-T-R-I-N-G. String. This is for protein-protein interaction analysis. Protein-protein interaction analysis. Protein-protein interaction analysis. Then we also have cytoscape. Cytoscape is for visualization of gene and protein interaction networks. Cytoscape for visualization of gene and protein interaction networks. When I was in Oklahoma State University, there was a lady that also came from Brazil. I was in the US for 9 months. She was in the US for 9 months. I came from Brazil. I went on football. She came on another program, another scholarship. And there are many scholarships in the world. Just the same way we are talking about this computer, this and that. There are many scholarships that. And what do I say? That even me, I don't know. It's when you search, how far you can search and you find it. So the more you can search on it, the more you can find. The same thing with this. All this we're talking about, if you just go and sit down, there's all the same thing you want to learn, you can learn from YouTube. Go and sit down with one. Just sit down with one. That works along with your own research. Okay, my research, you're doing research, let me even find out about this. Watch like, I'm talking about papers, 40 papers. Watch 40 videos on that thing alone. I bet you, if you don't change your research, come and knock me on the head. I'm telling you, if your research horizon does not change, when you see me, just knock me on the head. If you change it, 40 videos on just blast, how to blast, the correct way to blast. You will see things. You will see how to. One of my MSc students, who is in our second year, we're in your first year, in our second year, her seminar was, it was just born out of these things. Mistakes during blast. That was just a summary of our research. When people blast, this is the sequence. You've done all the PCR, everything, we look at go to the sequence. Then you now blast. When they blast, on the database, when you blast, there are many options of what the organism can be. So it's not just go to blast, let's give you one organism. It will give you many names, starting from 1, 2, 3, 4, 5, 6. On that page, you can get 20 names. If you want to continue, go to the next page, it will give you other names. And it's based on different criteria. So the usual thing is to pick the first one. That is not good. That's not how it should be done. There are some things you should look for before you select. The same thing, you will see that. So my thought, and this one is some paper we are writing now with one of my team members. Because the lady should not do justice to the topic. In the justice, what we are giving you a seminar topic, training is not just for you, we're playing. It's not to explain. Students take things for play. That's why there are only 2 of them. That's why you don't have notes there. You drop them and listen to jot. My notes are still there. Even BSc. They are still there with all the things I read, extra, that I jotted on the side. The same thing in MSc, I still have the notes. Shall the one that I aligns with my formula. So I gave her the topic. Doing 2 weeks to the time to present, she now brought this into me. Topic I've given you before you talk about seminar. So that I will have corrected and corrected, and then it will be a better work. So I mean, I don't fight students. I didn't know you before. So you learn that I was know me. So what's my role? I'm not the one that admitted you. It's you learn that admitted you. Then faculty gets a partner. So what's my role there? If anything happens to me, now that person will supervise the student. So I don't take things personal. I keep trying instruction. Don't focus. The student, good. If you have done that research very well for life, that is how you should be replicating it and getting better. I told her where to find this, where to find this. You have done the basic. If you do the first research very well, you do research very well. The next thing, go on. Now she didn't do it very well. I would like other people that want to, because you're supposed to do it very well, do the research very well, and then you can write something paper. Even the main call, I don't do it. Go and look for sequences, publish sequences in papers. On your own, blast them. They have said they are blasted and this is the name. You blast it and check if that is the name. Simple. Laziness download paper on that have sequences. Just go to Google. Paper that have sequences inside them from social year to social year. Even ChatGPT will give you the papers. Then you download papers, copy the sequences and blast it, and write if it's correct or if it's not correct. Like that. A day do 100. Between the time I gave and the time listen, a day does do 100. Or a day do 20. Like that, don't do like that. And we put it in different things. The one that is Apple, the one that's not Apple. And we put it like that, put it like that. And later you now separate them. At the end of the day, she said she's doing 30. Just look at 30. You know, is annoying. You have said things. You're not giving excuses. It's because of this. Most of them they use what do they call it? The name they call it. This one that they just instead of using the sequences, they put the accession number. Say it's accession number to use. They use accession number. It's the same thing now. If I use accession number, it's like I'm using this guy's sequence. Accession number is pointing to a sequence. And they give me accession number instead of sequence. It's the same thing. And but that accession number has a name. There is a accession number, you can put the accession number, you blast the accession number too. When you blast it, you also blast the accession number. It will give you the names. The name they now got in their paper doesn't align with the name you got from the blast. I wanted them to understand how they interpret blast with laziness. So if you are lazy, you don't go to the say, you are not go and sit down. But the moment you get it, you get it. It's computer based. And today, things are very easy. People just like to come to the lab, do those Indians that are saying that those Egyptians, 2 of them. One man one. Then they have English students, 2 again, one man one. Egyptians, they came from Egypt. That's what they said to Egypt. So when I was, some of the things I got, I asked the guy that should help me because my horse does not know those things. My horse used to push it for people that were there. My horse is using wet lab. They are also in environmental, but not using dry lab. So I begged the guy. In fact, I got a particular isolate, it was 88%. 88% black, very far from 100. So for that, you have to draw from the genetic tree. It was those people that I want to meet, the wife of that other lecturer that I want to meet, that guided me. So one of the lunch time, she came around, we sat down together, she put me through on my laptop, and then I got it. But the issue is that the moment you got it, you forget it again, except you're doing it continuously. So when I didn't get it, I tried to do it another time, I didn't get it, I called her again. Then she tried to explain to me. So apart from that one, that's how I got my phylogenetic tree. But today you somebody that got 100%. You got 100% too. Which phylogenetic tree are you drawing with me? It's showing 100% similarity, but I don't know phylogenetic tree. It's showing that it's highly similar. This one is far. That's why we got the tree. And if this one was alkali justifications. Alkali justifications. As I said, what the organism is doing is the key now, not just any organism. I travel with about 20-something organism. My home say, what are we going to do? Someone, what are we going to do with them? So you have to cut it to 4. The screen went down to 4. So those organisms, you're not testing them, we test it against one hydrocarbon. Before I travel, I was already testing against kerosene and petrol. So I have 24. So you can imagine just 24 organisms. Each one of them against kerosene. 1 against kerosene, 2 against kerosene. Then petrol 2, 1 against petrol, 2 against petrol. So when I got there, the first thing I did was to make a presentation on my work. I presented my work. At least you are coming from somewhere. You have to treat us your journey. So we have a lab meeting. We have lab meetings every Thursday. Two Thursdays. We have it this Thursday, very same Thursday. We don't have it next week. We don't have it after. So anything you have done in the lab in that 2 weeks, you tell us. So everybody will tell us. Sometimes they don't tell us. If you have had enough, then we read the paper. We will go and read the paper. We are reading 40 papers a week. If we want of them, we will share with us in the lab. And you'll be talking and see, and we ask you questions, see that you don't know. These things, I can, even if I die, I will not forget Zampoli. Zampoli I taught 2014. That was the first paper I presented over here. Zampoli for 2014. I traveled in 2015 because you have to do a recent paper. It was talking about hydrocarbons. Everything I want to do, these things are just for finished students. Students looking, they're not done. All these things I'm saying now, I don't know what the word means. All these committees, they can never hear it. I'm a PhD candidate. The man is teaching master's class. I will not attend that class because it's about environmental archaeology. I attend the class. I don't take exams. I'm not writing an exam. I just came for research. I sit down in the class. Every time I sit down. Everything, presentation, this, I present. In fact, there was a pair of presentation. I was the only one that presented. And the presentation was on cyber sequencing. He knew that was why, one of the reasons why I came, because of molecular aspects. So he pushed it to me, the presentation to do was cyber sequencing. The thing is, that thing is so simple. All these things are too simple. They are too simple. Just for you to just go through. I watched videos, I read it. I read different material, I watched different videos, then I understood it. Clearly, I prepared my PowerPoint and I presented. The man said I presented it very well. We went for conferences. I went for 3 conferences within 9 months. Here now, here for long, doing masters, you don't go for even half conference. Even half. Even you yourself are not interested in personal development. Don't talk about the lecturers. I went for 3 conferences. 3. 1, 2, 3. It was at the conference that somebody said, I was generalizing on arcanes that I only work on hexadecane. That is the purpose of the conference. One of the major purposes of the conference. We should criticize your work. If you are here and you're presenting your seminar, you will be sure the man hates you. You do seminar, the man says, you didn't put reference. You didn't do all of these things, the man said, you tell me to go and thank the man, buy him drink. You say, the man hates you, stupid man. You will not even greet him. The man said, just one comment. He said, I'm generalizing on arcades, but I only work on hexadecane. And we got to the lab. I said, see what this man said. He said, I'm talking about okay. So that was what I did work on cyclohexane and is it heptane or another one? That was that comment. We have our basis for working based on understanding, but that comment, yeah, is valid. So we went back. That you only enter the cave and then you realizing there are caves. That comment was what you took off from there. So that's about that. So yeah, we've talked about cytoscape of visualization of gene and protein interaction methods. We take that. Now let's look at the basic workflow of functional genomics computer analysis. Basic workflow of functional genomics computer analysis. Basic workflow of functional genomics computer analysis. I'm telling you, there's nothing you want to learn, and this area is cool area. I'm telling you, if you develop yourself in this area, just for you to come for PhD in this area, just take one research. All the data that you have in Nigeria, you just pick them one after the other. That you just have data, you take them to university, just take one after the other, put them together. Yes, I was talking about the Egyptians. Then I had one pseudomonas. In 24 hours, our setup we can finish degrading the something. In 24 hours. 24 hours, that's pseudomonas. So it was I don't know to tell about many stories about it.
Your work, because you know when I was following some of the things you do, and then I was wondering how that could help in areas that they have this oil degradation. Yes.
Yeah, I don't want to talk about Nigeria too, but I don't want to edit this video. Let's continue to. So let's look at the basic workflow of functional genomics computer analysis. Number one, data generation. Data generation. High-throughput experiments. High-throughput experiments. RNA sequencing, microarray. Chip sequencing. CHIP sequencing.
So are they the more implementation of data generation?
Yes, that is it. That's the first workflow. Under it, that's what we have. High-throughput experiments. Under the data generation, what is related to high-throughput experiments? Like RNA sequencing, microarray, chip sequencing. So this is taking it from the ground up. You have like that I said was in the US, that way that came from Brazil. Every time she is doing jet processes. Every time she's doing jet, what kind of experiment is this one? And what experiment was the big one? Every time jet shall be matching. What is all these experiments? So I get it now, this is what she was doing. Because me, the only time I run the gel was when I'm going to the molecular identification in my work. So I do DNA extraction. We didn't do DNA extraction, we do colony PCR. So I'm bypass DNA extraction. I just do colony PCR. So you use colony to pick. So they have toothpick in the lab, they sterilize the toothpick, and then they pick one toothpick, use a touch the colony, the 4-hour colony, and then pick it. And that's how you use, put it as in the PCR reaction. That colony, doing the PCR reaction. Then you run the PCR. That's what we call colony PCR. So you use the colony. Instead of doing DNA extraction, don't extract DNA. So don't do colony PCR. So the DNA inside the colony is what will be multiplied. We call millions. The answer that you want to gel. The essence of the gel is nothing. It's just to check if there's DNA in the PCR product. In the PCR. Because if you are not saying you do colony PCR, and then you say you have done PCR, then there's no PCR inside. They now send it for sequencing. Where is the money? You don't check for sequencing. You do the fluorescence. When you do gel, you see your band. You see the control, you see the everything. The one you are using, you label the first of the ladder, and like that. Ladder can come in any way. Ladder, then you have the organism 1, organism 2, organism 3. So at the end of the day, ladder can be here, this one can be here, this one can be here. So in fact, when they are moving, we will start, we put them from here, we drop them here. From the PCR, we drop them here, they set it up, then to move. When it has moved towards 80% of the movement, you go and stop it, and then you see that it's there. Then you remove the something, you wash the gel, you go and do you go observe it under the UV light. And then there's some UV light that have camera to snap it out for you. You have seen it here. You see it in the gel. With that gel, you want it to the machine to read it out for you. So machines are manual, some are not manual. So one are not manual, put it inside. If you take it like a photograph, like a television, you will see it. If you put it inside, you see it from outside, and you will snap it. And then you can have that. So once you see these are DNA bands, once it appears, that means your PCR, when you've done is okay, it has the DNA. So now you want to purify the DNA. You treat the DNA, and then pass something for sequencing. When you submit it for sequencing in the morning, before evening, you send the sequencing to the email. When you are there, will blast. That's all. But I see in this and Nigeria, they will blast for you. They will blast for you. You say, please, help me to blast it. You won't blast it, why will you blast half? Because it's a big deal. The person that you want to blast, you don't say rubbish. So you get the gist now. So if you now put yourself that, see, I'm a specialist in plastic, don't pay me for it. Advertise yourself all over Nigeria. You're saying that there's no job, there's no job. This is plastic. Yes. Is it some kind of software or machine? It's not machine, though. It's not just it's your phone, it's on your phone there. It's on your phone. Just NCBI blast, open it, now you see it. NCBI, you might blast it. So those who would think, is it that they don't know about it or? I just said that when you blast, it's like when you.
Okay. It's like an existing sequences of the blast.
Wait, this madam is heavy when she delivers something. Now when she delivers now, the father will name the child. The mother, she should have the name she wants to call the child. Then you that you are cosmates, you can say, master's baby. You to give him a name. I don't know, like my first born, the wife was working in school that time. My wife used to take biscuit and milk. So that's, when I figured out there are some things, and when I was working, it was me every month. So I just do the big bowl. So I look at it, okay, they do it in the office, I'll not be drinking it. So I'll bring it home. I will buy another one in my office or something like that. So you take biscuits and milk. So the one star of the work that are called the baby biscuit and milk baby. So you can see, the grandfather will name the child. Different people, neighbors will name the child. Different names. So you can have like 10 names. But which one is the main name? That is what blast is all about. Different names. Different names. Like my own name now, David, is my father gave me David. My mother gave me Benga. And my surname is Ernesto. So father, mother, there are other names. My father was so much activity. But that's my mother has a historical reason. Look at that means God raising you up. I think that means the warrior has come home. You are not using that one. You can see. But that's the name I'm coming to. You gave me, I told you. My mother gave me, but there are other names. I don't know if you get the gist now. So, in some places, they will say, no, it is the name that your grandfather gave the child and the child will bear. I don't know if you get it now. In some places, they say, no, it's the name of the woman, the mother that gave the child. The mother that gave the child. It's the name you gave the child without the hope. Some places say, no, it's the father, the name the father gave the child. No matter what. In some places, they say, no, it's the grandmother. Grandmother gave me, you are changing it with the stamp. So the same thing with the blast. There are different names when you blast for the organism. So that's why I said there are different conditions. You see different conditions, very length, everything. So when you now study all those things, you can now stand as a master. So that it's an open field. You can now stand and see this area, all over there, want to interpret your blasting to do. They will be sending it to you if you really stand properly. They will send it to you and you will be charging them. So after they pay for the sequencing, they will not pay you for blasting. Yeah. It depends on how you develop the software. Even they can send me as well all over the world, it's online. You will send them all over the world. So that's just, this one is just elementary, the other things, phylogenetic tree, people are learning it. Yeah? And then your LinkedIn profile, if you want to phylogenetic tree construction, contact me. Do contact and pay me. Don't wait for the local people here. I don't know how many people are being on the tree here. They will price it now. So you put it out there. Then you get paid. You get paid. The world is, the computer has made, that's why I'm everywhere. This one now, we turn this one now. Somebody will see this one now, that has saved his life. I'm online in different platforms, marriage, I'm there. Marriage and relationship, I'm there. I have to chat, I'm talking about that. I'm online because, see, those days you say, okay, I can sing, but the record label will not allow you to sing. They will not take you into the, they will not promote you. But now, internet is wide open for everybody. And I know you will have been watching some videos talking about pregnancy and some other things. True of course. So somebody that sat down and was talking about pregnancy, talking about policy, talking about this, talking about those things. So there are one ways. So everyone say that if you don't have a child and you want a child, you want a male child, that's how you do it. You want to, so people just stay, and people have need, and they pay them and they move on. So basic workflow of functional genomics computer analysis. Number one is data generation. High throughput experiments, RNA sequence. So the first you have to do the experiment. So if you come from the wet lab, it will come from the wet lab. You can't just start all this without the wet lab. I said that at the start. But there are a lot of things that are produced from the wet lab that are not analyzed. Yes. So you can just go on and take them and analyze them. That is your reason. In fact, what they call, the law is called copyright. Now, I don't know about some of these things for them. There's also copyright law. When you record a label or do a film, the copyright is not for life. For 50 years, 70 years, 80 years, or something, you can't work with it. If you record a film now, you do a film by like a particular film, because like Sonali's song that they did many years ago, if it has had 50 years or 60 years, based on the copyright law of Nigeria, it doesn't have copyright over it again. So you can take it and use it. You get it? You can take the song and use the song, and nobody can hold you. And that's what these musicians do. They go to old songs, all those songs. You can which song is in Orlando. Go and check his earlier song. They have listened to Orlando because they come from the same area. They have listened like this is basic method. Not direction, rather you can have indirect method. When their works, consuming their works. So when you appear, you wonder that did you train another person? So number 2, data processing. Quality control, trimming, normalization. That is data processing, quality control, trimming, normalization. And number 3, data analysis. So you see where that comes in. Statistical and computational analysis. Statistical and computational analysis. The first conference I attended, 2012. In 2011, I attended my first conference that I didn't present the paper. I attended, somebody just invited me to come attend to have the experience of our conference looked like in 2011. That was the year I became a lecturer at the University. I became a lecturer here to that year. So January after the University, October I was here. So the guy just invited me to Joseph University, owned by the CC, I think he around. So I went there. I stayed in his lecture in last school. Just in practice all around. His lecture in last school, so he invited me to come and just see how come. I said, oh, let me just come. I was ad hoc LOC registration, registration. I was within the registration person. Then in 2012 was our Moringa conference in this university. And Professor Akinyoju, you know him? Professor Akinyoju was here before. Okay? So he gave some students a project that he retired. So the students were already working. So when I came, he just said, okay, you take this student. That girl is a doctor today. Because she, after PhD, she went to university. So she did some work on moringa leaf, extract, and antibiotics. Because these days we still have students these days, it's not enough, students will understand. So she got the isolate from teaching city. Because she did moringa, we didn't follow all the process. The process are very simple. Don't follow the books. We have to have a protocol. Just follow the protocol. Understand how you have to study. Understand the protocol. And just follow this. Follow the protocol. We get an extract. We apply it. 24 hours. There's no this thing. Okay, let me for this and nothing. So she repeatedly, she did everything, nothing. Okay, was recording the values. 0, 0, 0, 0, 0, 0, 0, 0. Where did one ask them? Where did they take the isolate from? One says from Move. Move. You know, told me move because it was general. Move the anti-capo. Something in the normal, stop my group. So that was how I was saying, okay, go and collect everything from our lab. You just release some pipe, don't have any cushion. And that's, you know, I tell my students, my research is not, I don't know where we are going. I just say, let us check and see. Based on my studies, let's just check this and see. I said, go in university, I will not rank off, based on what we get. So our focus was hospital isolate. But you are negative. But their results, I said, okay, we are doing the right thing. We check again, we are doing the right thing. And that we made it and test students. If you are not doing the right thing, any other thing, forget it. Just be recording it. You may be detecting something that most of us don't want to get positive results. Everything is what is result. We can understand this. So we don't give it all negative from the clinical isolate. With the same thing, the same molecular we don't use isolate from here, which are not clinical. Come and see zone of inhibition. So before somebody have wound, normally if you have wound, you don't even go to anywhere, just leave it to go. So this one didn't go, they go to, they'll use drug, but they didn't go. You now want hospital now do swab again, now culture it. Hospital now have the culture. And that's what we want to go to. So that's why it's stubborn. So, data analysis, statistical and computational analysis. I was talking about the conference 2012. So that was the result I think I think they for 2012 conference. That time I didn't know about statistical analysis. Statistical analysis. ANOVA. I think I did ANOVA in 2011. About statistics for biological sciences students. It was on paper. They were using computer. You know it. So I just do my table, all these values I have, plus, minus, this one 0, 0, this one 25, this one 2 centimeters, like that. I just write everything like that in the table. So one guy came and he looked, he said, there is no, you didn't do data statistical analysis. I said, I did it. I said, look at it. Table last one, go, share anything, so for that, I said, look at it, I did it. The guy said, didn't do it. I said, I did it. Look at it. The guy said, this is not what are you saying? This is it. This is table. This table is never not. I didn't know, and I was ignorant. I was telling this. after the test, it was a one-day conference. So I came from Oyo, my family was in Oyo, I was here. So I just drove in because it was during the holiday. I just drove in after the food and drove back. So I now went to sit down. This guy was talking since. And I mentioned only that discussed here in the surgery's office, in the office. I mentioned only the surgery. I said, see how you're arguing. This was how I was arguing that. I said, you did this and that. Go and check this and do it. And now you now check the second combination and see that you have done plenty. Everything data analysis. So like this 25, that's to be plus or minus something, A based upon something. Showing that okay, this one is A, this one is B, this one is so at a spot you will see it across the line that okay, this A, this B, different from this. The same this data analysis, because I said it, one guy just went. He just went. People are funny. That's why I said that students what you should do. You should compare all the sequences and check the correctness. If you do that, it's very. I'm not angry, but I'm just saying, I'm not angry. That's just the person thing. We are still good friends. We are moving on. I just saw this one 2 days before she goes. So because I thought Kule that one of those Kule did it, one guy just come like that too. And he did to the second data analysis too. Then we have 10. This is for A, this example. 11, this is for C, 12, this is for D. 13, this to power A has C, 13 this to power B. You to look at it. 10, after 10, what do we have? 11. So why would between 10 and 11 is 10 if 10 and 10 is equal to 11. So if you are going to have similarity, it should be between A, this should be A, and this should be what? B. Why is the B now in 13 and D is in 12? That's why the guy put everything like that. I now called him and said, wait, I pointed it out here. I said, what is this one? You didn't analyze for this one. He said, yes, this is actually. And the one that said she didn't analyze now, I was mentioning data analysis. In the case there, I was not saying that data analysis, everybody see, you said that the one that is 10 is just about A, then the one that is 15 is B, then 11 is a D, and so you don't get 10. No maximum 11 maximum, then sometimes you have 10.5, 10.7. So, in fact, sometimes you have AB. Sometimes you have AB. I mean that we have another one now that is 10.2. So 10.2. There's mistake here already. The way I write it. Because this level is supposed to be B, not this 13. Because 10 and 11 are close. So 10.2 will be what? AB. That is, you are not the one that will write it to easy. I know what I will do it. That one is going to make money. One table is 1,000 that time. And now I'm doing that way. One table is 1,000 that time.
So I write it in 1,000.
Yes, to do a number. It doesn't take me, if I sit down with it, doesn't take me 10 minutes. It's not that just look at it and doing it. It's just all data, just get it to duplicate. Take it to Excel and do this thing there. Carry it, run the analysis surveillance. It's a software, ANOVA software. I run the ANOVA SPSS. Then I open it, I run it, and I always value the way we should put it in Excel. I just carry them, put them inside the ANOVA. It will analyze them. Then all these things are inside table already. On your table, you lost the table. Now I have found the mean. The mean is there. You understand? The mean will be in the table, ready for the rest. So if the mean is in the table and you want to use, you run data, I take it to Excel, and then import it to my ANOVA. Ask the one, if you pick it up from there, refer to my ANOVA to interpret it. So all these differences, you know me, I found the mean. ANOVA will run it and find the mean and now give you where all these things will be. So it's not you that are going to put it there. But after ANOVA have done it, all you need to do is just come and put it there. Look at it in the ANOVA result and put it there. That one you have to take time to interpret it and put it there and put everything there. After putting it there, you will see it. There was one girl that the master's supervisor. So after they do that, I work, they said, "You don't print the dissertation, you want to print it." As he said, he reached the place, he said, "Well, I just took it out. Threw it away, fling it at her. She found them always crying. And he's a married woman with children. He was crying. You know when someone is crying, I'm going to cry inside jail. I said, "Sit down, what happened?" You know, I said, "Yeah, you know, I said, that my jail, you don't see jail, you're one, you jail two." You cried one day, I will say, "Which kind of cry for this one?" Okay, what happened? You see, I didn't do, it's one that I should not pray to. I don't want to pray now. It's I didn't see what you know, break it, see this or this. I said, this is simple, I can do this. I need something to do, so I'll look opportunity. It's not this first opportunity. That's how we try to train people. And they can think, I'm just okay, someone can do it. Go and meet that person, that person can do it. The most good at is to push up to now. I'm not ready. I can't imagine when I'm doing an over, doing all these things. This is 5K. The fullness I think for a high level for some people. See this one of these things. We still write, we write for people and other things. I wrote for somebody last year. I had a 50K. I write something for me. I don't think they will in 5 days it was ready. So in 5 days, please God. So in 5 days it was ready. The man looked at it and said, this thing, no mistake, nothing. Give me another one. So this one, I sent to a day here, I went down. I don't know what the day, in fact, there was one that gave me 100. Sent her along collect like that. Yeah, my wife. Yes, so the table will change. Spend the money on do the work. So that was that. Let's go to the next one. That's data analysis, statistical and computational analysis. The other one is statistical analysis, it's also about computational analysis. That was exactly what I asked her to do. There are mistakes. You go and check a lot of, that's why you should read papers. You see some data, you want to go and learn that thing now. Use it in your own work. Your work will come out very well. We sent one paper to Notolue Biologics. It's a free journal. In Biologics, the journal in biological sciences. Romania, in one university in Romania. The guy that is the editor-in-chief was born in 1982. So I didn't see you around. The problem is that if it's here that such people are. So this journal is not just joke, it's part of the good work of the one. So I just sat down. The next one that I now say with somebody again, we sat down, work on it very well before we sent it. So that's the next one, and the basic work is functional annotation. Assigning biological meaning to genes. Assigning biological meaning to genes. Number 5 is pathway and network analysis. That is understanding gene interactions. Understanding gene interactions. Number 6 is biological interpretation. That is drawing conclusion from analyzed data. Drawing conclusion from analyzed data. Drawing conclusion from analyzed data. 5 is pathway and network analysis. That is assigning biological meaning to genes all the time. Okay. Now let's look at the databases used in functional genomics. Databases used in functional genomics. Number 1 is NCBI gene. And GEO and GEO. NCBI gene and GEO. GEO capital letter GEO. Gene information and expression datasets. That is what this is called. gene information and expression datasets. Gene information and expression datasets. The next one is KEGG, capital. Pathway and disease associations. This is for pathways and disease association. The next one is UNI, capital U, NR, capital P, RNA-seq, UniProt. Protein sequences and functional information. Protein sequences and functional information. The next one is Ensembl, ENSEMBL, capital E, small n, small s, small e, small m, small b, small l. This is for genome annotation. Then we have gene ontology consortium. That's the next one. Another term is gene on top, not on top, ontology consortium. This is for functional gene classification. Functional gene classification. Now let's look at application of functional genomics computer analysis. 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 Number one, identification of disease-related genes. Identification of disease-related genes. Number two, drug target discovery and development. Drug target discovery and development. The next one is understanding microbial metabolism and adaptation. Understanding microbial metabolism and adaptation. The next one is cancer genomics and personalized medicine. Cancer genomics and personalized medicine.
Personalized
medicine. The next one is agricultural and environmental biotechnology. Agricultural and environmental biotechnology. Biotech. Advantages of computer analysis in functional genomics. Advantages of computer analysis in functional genomics. Number one, handles large-scale biological data efficiently. Handles large-scale biological data efficiently. Number two, enables accurate prediction of gene function. Enables accurate prediction of gene function. Number three, facilitates system level understanding of biology. Facilitates system level understanding of biology. The last one here, reduces time and cost compared to experimental only approaches. Reduces time and cost compared to experimental only approaches. I'll explain that when we are talking about COVID-19. Using time, every time that you have time to carry out the analysis. COVID, people were dying in China, in the USA, every day. And there was no time to start checking and checking. So you have to use the regular document. So you use the regular document to check whether the data is so you have to use it. You don't need to go and use animal models. And in fact, in some countries, animal models have been stopped. All this one that you do something, you're not going to use mouse. Some countries they say no, don't use them again. So you shouldn't use them again. Limitations and challenges. requires computational expertise. This is the limitation based on learning. If many people see it as limitation, but you learn it, you have the edge. You have the edge. 3 days ago, I was here till around 7 p.m. I was doing some things, personal work. There are some books about wealth. Think and grow rich, tough time never happened to you, millionaire mind, that. I don't have time to read. I've read some of them, all of them before. I don't have time to read them now. I don't have time to read them now. That's why this is the time you have to read. Read like no man. I read like crazy. Somebody knows something that I said, what happened? Saw me at Block 3. That was our apartment then, Block 3F1. That was where we used to, most times, we used to meet there. I was on Bio Boys, Biological Sciences, CRISPR. Now, I just see that everything has changed. I used to write about Boys. I used the nickname Big Daddy. That's why I write about the missing bar. I wrote about Archimedes. Archimedes ran out of the back of naked. When he started to make and make. You know this story. That's why I wrote about the naked scientists. Naked scientists. You see this naked on the back of discovering nothing. Archimedes, at least, he was one that found the Archimedes principles. So when he found it, he was in the back top. He found it that he put something, he said it's coming on. He said, this thing I need to have found it. You know, I this guy in the back to the asylum. So from there, that's the naked scientist. Many things are getting naked, discovering nothing. Archimedes discover something. So I wrote many like that. Now there's no pressure of the students again. So what I did that day was all those books I wrote to ChatGPT, I said summarize them. I use the book, summarize it. So summarize it, 1 page. I keep it. Another one, so I was not doing that, and many other things. I still want to. So you know what I've read before? Little tip now, I find out. I don't need all the story. I know all the story. I just need to get the listing. I'm sorry, I talk about church now. Anytime somebody is preaching, I take the decision, I know the person prepared everywhere they're going to preach. So when the person shouting, woman gets in the bottom song. Well, the meaning is but that one. If I need to record something, I get where the person when you make business, I know it. So it's not all the 1 hour that they use in the WAS that is really carrying me. The way carrying me, I don't know. The way the person will talk at that point is different. Even the person will listen to the recording, he will say, I wouldn't know what I said. So that kind of moment, I really should know it from experience. I will know it. Just be using baga, you just see that power voting law, and something inside this thing like this. What the person is saying, getting inspiration of another dimension. Your head is saying to yourself, interpreting different things. The same thing happens. So require computational expertise, then data quality and annotation. Data quality and annotation errors may affect results. Data quality and annotation errors may affect results. Data quality and annotation errors may affect results. About this course we're talking about now. Part of what you guys should have seen is that this sequence, was it done using 1 way or 2 way? There are some that will say 1 way, some will say 2 way. So 2 way uses the forward and the reverse. 1 way uses only 1 way, and then they use the complementary to complement. But 1 way, we give you all this, you can find the complementary place. There's a pair that's complementing A, another one complementing C. So when you go one way, you cannot find the complementing way. The way there's error, if you're one way, there's error here. Then continue. There's error here. But if you do the two-way, this error now, the two-way catches the other side of this. So you can now, this complement that you have here can now give you what is here. This other complement that you have here can give you what is here. You get it now? So those are the things. So you're going to shape, these are likely things. Now, interpretation often needs experimental validation. Interpretation often needs experimental validation. Interpretation often needs experimental validation. Yeah, the one of them. Functional genomics, computer analysis integrates biology. That's the last part of it. Functional genomics, computer analysis integrates biology, computer science, and statistics. Integrates biology, computer science, and statistics to uncover the functional roles of genes and proteins. To uncover the functional roles of genes and proteins. True bioinformatics, true bioinformatic tools and databases. True bioinformatic tools and databases. True bioinformatic tools and databases. Researchers can analyze complex data sets. Researchers can analyze complex data sets and generate insights. Researchers can analyze complex data sets, identify biological pathways, and generate insights that advance research in medicine.
Complex data sets.
Identify biological pathways, complex data sets, identify biological pathways, and generate insights that advance research in medicine, agriculture, environmental anthropology, and environmental science. Environmental anthropology and environmental science. See, I knew that before the docket traveled, we were discussing getting our own kind of database. You are talking about something like that the right time. People that have a high, that their brains are open. They don't remain around. You go away. You don't remain around. You're gone. There are many opportunities. We were talking about we having our own like badges manual where we can put online. I mean, databases online that you can go to do the traditional this thing, all these tests, the use of tests. Those days you have to go and know about this manual. Now don't go online. I do this test. What did this say? To give you the results and to give you the reference. Some people say, I don't want to use ChatGPT with this. Don't use it because of business. If you don't know anything before, say you use it, you do what you should do. You don't know anything before, don't do anything. You're not going to ChatGPT. You will see do rubbish to show you more rubbish. There was a paper that somebody published, and we will see the chief editor. Yeah, I do not have the time. Somebody was showing me one of the journals in the university. If guy used ChatGPT, you have to clean it up, after you use ChatGPT, read it. When you use ChatGPT saying, if you want me to give you more this thing, I will give you. Can I do this? I shall do this. You have to beat here, but this and that. You don't put that, so send it to them. Don't do that now. You know what the students are.
There was a newspaper from, I think, Pakistan.
Okay.
The person was also from ChatGPT. Yeah. It was there.
Yeah. That one is this one put Oh, yeah, he said brackets. You know, that kind of equation, it's supposed equation brackets, something. ChatGPT does not know that it's just supposed to just number bracket, dot, this and that. And for you to wrote it, and you to left it like that. So the guy was showing me. I told the guy, I said, no, ChatGPT show ChatGPT. You clean it up. ChatGPT is a tool. Use it. But you should know what I'm doing. To go to ChatGPT and say, okay, I use it a lot. I said, okay, I want to write these references, give me references. ChatGPT will give you wrong references. It can give you the story, but it will give you wrong references. You should go and copy and not use it. You see that it's wrong reference. And there are some very strong journals. Google reference, they will check everyone. And they'll tell you that what they wrote in this reference is different from what you said. It now is the name of the biology study of science. But the reference you put is not about chemistry. So you will not know if you don't check it. Just put biology study of science, then you put Ababio chemistry, or Ababio something, Ababio 1997. Then the guy will now go and copy the Ababio 1997, put it on Google, and say, yes, you talk about chemistry now. How do you now say it's biology study of science inside chemistry? You say, no. Some editors and analysts, it's very simple to read. They will take all your references and check it. Now let's look at assess the sample questions. Number one, define functional genomics and explain the role of computer analysis. Define functional genomics and explain the role of computer analysis. Define functional genomics and explain the role of computer analysis. Where's your phone number? Define functional genomics and explain the role of computer analysis. Number two, list and explain 4 types of data used. That's your WhatsApp. Yes, sir. I'll send the material to you. I will not send this one, but I know you will share it with your class. Don't you like go and share this one? I won't send. I will look this one, but I want to send it. They will not see this one, so I will send you another material. If you like, go and tell them if you want this material that you have lecture. Just send the number to them that this is the lecture and you not send it. So they will take time to read it very well.
What types of
List and explain 4 types of data used in functional genomics. List and explain 4 types of data used in functional genomics. Number 3, describe the steps involved. Describe the steps involved in functional genomics data analysis. Number 4, mention 5 bioinformatics tools used in functional genomics and their functions.